So as I mentioned in the outline of my presentation, the first part, I will look into the pathogen causing root rot. The symptoms you are probably well aware of, but I think it's always good to re-emphasize in a way how it looks like and why it looks like. And then the life cycle and the epidemiology of the disease. And then we know about some management practices and how they affect the root rot. Crop rotation is one very important point here. And then cultivation and varieties are also important to consider as measures to combat. And finally, I would conclude on messages. What can we do about root rot in the field? So the pathogen is a fungi, and the disease is a soil-borne disease caused by a fungi called Rizoctonia solani. And this fungi exists in different strains, and these fungi have a special feature which is called anastomus, because anyway, the hyphae are able to merge and exchange genetic material. And by this, of course, the variability of this fungi is quite huge. And of course, the potential to infect sugar beets is quite large as soon as there is this fungi in the soil. So all the different Rizoctonia strains are differentiated by their host plants, and several of those strains are capable of infecting sugar beet. Basically, we talk about two strains. There's this group AG4, and this is already exclusively associated with early outbreak of damping off, which is shown on this image here below, because this is also Rizoctonia disease, but affects the plant early in the season after establishment. So the base of the plant becomes dark, and then the plant in a way wilts and in a way dies off. So that's what we call damping off. And of course, this affects the crop stand and also yield finally. But the group which causes root rot and crown rot is the AG22, 3B. This is the main pathogen causing the disease in sugar beet. And under the microscope, it has this typical architecture of the mycel, of the hyphae, because in a way on the joints, the joints are rectangular, which is very typical for Rizoctonia solani AG2. So if people look at the pathogen in the soil, they will look for this kind of pictures to identify that the reason for the disease is really Rizoctonia solani. So in absence of a host, meaning sugar beet or other host plants we will talk about later, this fungi forms so-called Sclerosia, which is a kind of packed mycel, and this gives them a survival structure. Or this mycelium can also survive on plant debris, on residues of different crops. So we got to know how the fungi looks like, but of course, with just without a microscope, in a way, we don't see this fungi in the soil or on the sugar beet. We see the symptoms, and the symptoms matter for the damage caused by this disease. So even though the infestation starts in the root, as we all know, quite often the outbreak of the fungus is observed first on the leaves, because as a consequence of the root infestation infection, we have symptoms early. In a way, the outbreak is early in the season already, but we don't see immediately the effect of this disease because it takes quite a while before, in a way, the mycelium in the root spreads and grows until we see, in a way, the symptoms on the leaves. You probably have seen also in your fields. So, and even though the infestation is already early in May, June, quite often the disease becomes only apparent late in summer when the leaves in patches start to wilt. And the typical pattern of these patches is that it grows along the roots because the fungi spreads very much along the roots. And then it grows gradually in the field to other rows and so forth. So first symptoms is the wilting, sudden wilting, and then the leaves in a way get chlorotic, and further on necrosis is seen. And but the leaves stay at the plant, and then we get a kind of star-shaped picture from a plant. All the wilted leaves are in a star-shaped pattern around the beetroot. Also, dark brown spots can be observed on the leaf, on the petioles, on the leaf base, so to say. Yeah, as I said, the foliage remains attached to the crown, so it's not fully decayed and cut from the plant. So as soon as the soil reaches a soil temperature of 15 centigrades Celsius, exudates from the sugar beet plant activate the Sclerosia to emerge, to germinate, and to generate a new long hyphae and mycelium. So the host plant, the presence of the host plants really triggers, in a way, the fungi to grow, as it's the case for most soil-borne fungi. And the fungi infest the root at the part where the cell walls are not as strong. So that means the kind of new developing tissue at the root tip. So, the first symptoms you normally see at the tip of the beet or the tip of the pivotal root in the soil. And from there, the mycelium proliferates in the root, and it takes a bigger part of the root. And as a kind of secondary infestation, you have all kind of fungi and other microbes, which in a way uses the energy and nutrients of the beet tissue to grow and to propagate. So first you have the infestation, the fungi takes a big part, and then you have a secondary infestation with all kind of different fungi and microbes. So, and then at a certain point in time, the mycelium reaches the xylem, which are the vessels which that transports water and the nutrients. And as soon as the fungi is in these vessels, then the vessels are attacked and destroyed. And that's the point in time where we will see the first kind of wilting symptoms of the leaves in a crop stand. In absence of any kind of analytical service and really methods to identify whether any kind of disease is really caused by Rhizoctonia solani, There are a couple of deficiencies and diseases which can be confused with Rizoctonia solani. A well-known one is the deficiency of boron, which also causes the hard and dry rot in the leaf, in the center of the leaf, where the new leaves are developing. And that is just because boron is lacking there, and the tissue cannot be developed properly. Cell walls and so on are very weak, and then you get also infestation by all kinds of different fungi, which causes something like hard and dry rot in sugar beet. Also, a nematode can affect the leaf tissue, Dithylenchus dipsaki. It's a nematode living on the soil surface and can affect the leaves. Can also cause some root rot, or not root rot, head rot or crown rot on the beet head. And then there's another, is a scab, Aphanomyces cochleoides, which causes also bulk-like growth on the beet body. Well, I think it's not too difficult to differentiate these diseases from Rizoctonia solani as soon as you look at the root, because all these three deficiency or diseases normally affect only the leaf part and not the root part. When we look at the geographical occurrence of root rot in Europe and US, this disease seems to be widespread now in the main areas where sugar beets are growing. And the sugar beet growing, at least in Europe, has been very much concentrated in certain areas because the sugar mills became bigger but fewer. And of course, the logistics of sugar beet makes it necessary and as a prerequisite that the beets are grown close to the mill. And we see that in those, and that means, of course, sugar beet are grown more frequently in the crop rotation. And as we will see later on, anyways, the rotation and the number of different crops in a rotation and the crops grown in a rotation are very important for the spread of Rizoctonia solani root rot. So, for instance, in France, on the left side, we see two areas, in the northeast and very much in the center, where the soils and the weather is appropriate for sugar beet production. And we see that in those centers, we have a strong presence of root rot, and there are some others in the west, northwest also, where in a way the presence is limited, but maybe it's just a matter of time before we have also more severe problems in these areas. Because the threat in a way grows with the, let's say, inoculum in the soil, with the availability of fungi, hyphae, mycelium, and so on, and this accumulates over time. In Germany, also the same picture, two main sugar beet areas, one in the west around Cologne, soils probably a bit similar to Ukraine, silty soils, very fertile soils as we normally have for sugar beet production. And also here we have a kind of strong presence of Rizoctonia solani. And the other one is very much in the southeast, Bavaria, another big area for sugar beet growing. And also here, root rot is a severe problem. Also in US, you can see the main sugar beet areas, and also there is a kind of threat from this soil-borne disease. Different to France and Europe, and I will talk about later, is that in US they have a fungicide, and they are allowed to use a fungicide, and this one seems to have some good effects under conditions where we have really a very high severity of this disease. Of course, for this widespread of this disease also causes some economic importance and of course economic losses, and this can be only estimated based on some research going on. So the impact on beet yield of course depends on the area and severity of the outbreak, but losses of recoverable white sugar as soon as the outbreak and the symptoms are visible in the field are assumed to be in the order of magnitude of 5 to 10%. But also, if you have a very severe infestation, 50 to 60% of losses are not uncommon. And in France, the French research found trials where even 100% of the yield were gone. Belgium reports a figure of 45%. In the Netherlands, 25, sometimes even 100%, and in 60% in Germany. All these institutions which have observed this kind of very high losses are institutions for sugar beet research. And I used quite a lot of information from this institution to compile the information of this session. And the research has started in the 90s, late 90s, and continued up to date, but most intensively in the early 2000s in these research centers, and they looked into a couple of factors influencing the disease. And most of the results I will share later are based on the research of these institutes. Of course, the beet yield is one thing, but of course it has also an impact on the, let's say, the quality of harvest quality. And in sugar beet, that's very much the recoverable sugar. If we have an infestation, we have a severe reduction in sugar content. Also, as a kind of indirect effect, more soil sticks to the beet because it doesn't have this smooth surface anymore, and you bring more soil into the meal, which is also very much unwanted and affects quality and so on. Also, the recovery goes down because a higher level of sodium, potassium, and amino nitrogen is in the beet. And then, of course, some of the beets need to be stored either in the field or in the mill. And if you have a rot with all kind of organism causing this rot, of course, this rot can spread also in the piles of beet, and it can affect quite even bigger part of the harvest than the one which is, let's say, directly affected by the root rot. But before we go into factors which promotes the spread of root rot, or those factors who can suppress the root rot, let's have a bit closer look to the life cycle and the epidemiology of the fungi on the sugar beet, which is illustrated by the next slide. So as I have mentioned, the starting point is always the inoculum, means Sclerosia or mycelium on plant residues, which propagate, or in a way, which start to grow as soon as a host plant is in the field, and they get the kind of trigger from some compounds the host plant exudates that makes the fungi grows. So they grow into older mycelium, and this mycelium colonizes the surface of the root and in a way grows further on the root before the mycelium develops what we call a kind of very dense pile of mycelium, what we call a infection cushion. And this infection cushion develops enzymes which breaks the cell walls of the host plants, the sugar beet root and beet, and then the hyphae of the Rizoctonia fungi penetrates the root and beet tissue quite a lot. And that's the stage when you can see the first symptoms of rotting. And of course, as soon as you have host plants, the inoculum grows, and the quantity of inoculum in the soil grows with, let's say, residues left in the field, and then again, in a way, the entire process starts with overwintering forms of the Sclerosia or mycelium on straw seeds and so forth. So this is the cycle of the fungi in the soil and the involvement of the beet, but there are also other plants who can host Rizoctonia solani, AG22, and also those host plants can contribute to increasing the inoculum of Rizoctonia solani in the soil. When it comes to the factors who can suppress the inoculum, meaning in a way decrease the number or the quantity of hyphae and Sclerosia mycelium in the soil, this could be in an extended rotation with a 3 to 5 years gap between two beet crops. Cereals seem to have also a suppressive effect on the development of inoculum. So additional wheat or barley crop before sugar beet, meaning ideally 2 cereal crops before sugar beets could help. And then also it was found that intercropping by, for instance, brown mustard or oil radish can reduce the inoculum. Quite obvious is that Rhizoctonia tolerant varieties, where the fungi doesn't have the same kind of opportunity to grow and thrive and to propagate, contribute to reduce the inoculum, and the weed control of, in a way, wheat plants which can host fungi has another effect here on suppressing the inoculum. When it comes to conditions under which the fungi can thrive, soil weather conditions matter the most here. I have mentioned already that the fungi starts to grow at soil temperatures above 15 centigrades, and in a way the conditions are more ideal if we have very high soil moisture in May and June, when normally in a way the infestation will start in sugar beet. So temperature above 15 and abundant rainfall, a lot of rainfall would be a kind of, would promote the incidence of root rot. Also, poor soil structure and soil compaction means that there's very low a shortage of oxygen in the soil, which is not good for the crop, weakens the crop, but strengthens the fungi. The fungi likes more anaerobic conditions. Something we quite often have also in heavy soils, so very heavy clay soils can be prone more to root rot infestation. Poor drainage and waterlogging, so everything what has to do with in a way soil moisture and excess of water in a way promotes infestation. And quite often when we have a good harvest of the previous crops, there are mountains of residues to be incorporated. And if they are not evenly incorporated, also these pockets of straw in the soil is a starting point for the infestation with root rot. When it comes to conditions which suppress the growth of this fungi, it's obvious that in a way low temperatures in May, June, when the sugar beet starts to establish a root and the beet reduces the infestation, good soil structure and soil aeration. It's obvious as the opposite to in a way excess of water. Light soils with good drainage, evenly incorporated harvest residues, and adequate nutrient supply and liming, meaning everything what makes the plant stronger in a way weakens the fungi, except in a way the temperature. Of course, the sugar beet and the fungi like higher temperatures in soil to grow. So this is the only thing they have in common, that they grow better with high temperatures. So yeah, then let's continue. So in the first part, we have seen now what kind of factors promote and what factors suppress the outbreak of the disease. And in the next part, I'd like to come to the impact of different management practice affecting these different factors. So the next couple of slides are very similar because they come from, let's say, the same source of research. Next to each other, we have the disease severity, and I will come to that, what the figures mean, and the corresponding white sugar yield of sugar beet. And in this research, the effect of susceptible compared to resistant sugar beet variety has been tested in different crop rotations, which were maize wheat, or wheat oat before sugar beet. So the disease severity, the scores for that you can see in this table. One means in a way hardly any surface of the root with visible Rizoctonia lessons, and the highest score, 9, means 75 of the root surface is with dry rot cancer. So this is the meaning of this disease severity. There's a kind of index system to measure the severity. Yeah, the results show that in a way the disease severity was between 2 and 4 in these observations. 2 means less than 1% of the root surface visible with Rizoctonia, and 4 means 5 to 10% affected. And the infestation depends, as I said, both on the variety, whether it has been a susceptible or resistant variety, but it depends also very much on the crops grown in the rotation, where the 2 crops, maize as a host plant was in the rotation, or whether wheat after maize was before sugar beet, or whether we had 2 cereal crops before sugar beet. And as we can see that a resistant sugar beet variety doesn't respond to the effects of the rotation. So there's no significant difference in severity of the disease, But when we have a susceptible sugar beet cultivar, there's a quite decrease in disease severity with the number of maize crops grown in the rotation. So maize in the rotation has a significant impact on the severity of disease. When we look at the white sugar yield, the one which most interests us the most when it comes to economy of the crop, and if we have a resistant crop, the response to the rotation is very weak. There's no significant difference in the yield, even though there's a trend towards high yield with 2 cereals crop in a rotation. But if we have a susceptible crop, then there's a significant difference in white sugar yield, depending on the number of maize crops we have in the rotation, which of course indicates that maize as a host plant propagates the inoculum, and the sugar beet, if it's susceptible to the disease, reacts and responds to this higher inoculum in the soil. In conclusion, this means if we have a very high pressure for root rot, we have to, or it's recommended to use root rot resistant variety. And as a kind of additional measure, if possible at all, reduce the number of maize crops in the rotation, and maybe substitute with the additional cereal crop. This is a very busy slide, but it shows a trial where different maize genotypes, varieties were tested in the field, and the disease was rated on the maize roots. As I told you, Rizoctonia solani AG22 also affects maize roots, and the disease severity depends whether in the trial they have applied additional inoculum of the Rizoctonia fungi, or whether the trial was done just based on the natural inoculum potential. And you can see that different maize varieties react in terms of disease severity mainly to the additional inoculum, but also when you have a kind of natural inoculum, there are some maize varieties which build up the disease more intensive than others. So when we look at the correlation of the yield, the white sugar yield on the y-axis and the kernel yield maize on the x-axis the year before, and look at the different genotypes or varieties, you see it looks like there is a kind of linear correlation. That means in a way yield maize variety which yields high, we normally would expect also that the sugar beet grown after maize yields high. There are a few exceptions like the variety 8, which means that one performs still very good in maize, but is awful in sugar beet, and the same applies for variety 5, which performs, of course, low in both in yield and in maize. So to conclude, maize genotypes differ in their susceptible towards Rizoctonia solani. Means they have the different potential to accumulate inoculum, and this inoculum potential seems to affect also the maize varieties, and the maize genotype grown before sugar beet influences also the sugar beet crop. So in conclusion, harvest residues of maize genotypes might differentially influence the survival of Rizoctonia solani in soil. And if we know about the correlation between this and the potential of building inoculum of different maize varieties, we can also reduce the incidence of root rot in sugar beet. So we learned that now the infestation has a lot to do with what happens on residues and the residues of the previous crop. And this indicates that maybe the tillage, soil tillage system affects also the incidence of root rot. And the effect of the cultivation method is shown on this slide. We have basically 4 different cultivation methods compared in these 3 different rotations. So again, we stay with maize wheat, and wheat oat before sugar beet. And the cultivation were to the crops before sugar beet, a plow and then just a cultivator. Number 2 is plow and plowed for the sugar beet crop. Number 3 and 4, both the residues were plowed in, and the difference between the 2 is that for the sugar beet crop, as it has been only cultivated with a wheat swing, or with the previous crop has been, the residues have been plowed in. When we look at the disease severity, it looks like that in the maize, wheat, sugar beet rotation, the effect of the cultivation method is there's no significant differences. In the wheat, oat, sugar beet crop, it looks like a low tillage system before sugar beet could reduce the severity. And this has probably something to do with the problem that if straw needs to be incorporated with a plow, we have this risk of layers, mattresses, as we call it, with a straw, and this can be avoided if it's not plowed. So the residues are more on the surface and cannot contribute to the growth of the Rizoctonia fungi. The most significant effects we have in the maize sugar beet rotation when it comes to disease severity. And here it looks like that plowing, if the maize crop is just before the sugar beet, plowing is mandatory. So with plowing before sugar beet, we can reduce the severity quite significantly, and correspondingly also the yield in a way is higher after the land has been plowed compared to in a way just cultivating surface type of tillage system. I think for the other crop rotations, it's a kind of very indifferent picture when it comes to the yield. So either one of the different cultivation methods seems to be appropriate to get, let's say, the highest sugar beet yield. The next slide shows you the effect of nitrogen nutrition and the preceding crop in sugar beet. We have the response to 4 different nitrogen rates applied to sugar beet. Either the control without 45, 75, and 112 kilograms of nitrogen. And the graph shows you the response to these different nitrogen rates as the sugar, white sugar yield. As we know, P is a very good preceding crop, leaves some nitrogen in the soil. And with this crop, on this site, only 45 kilograms of nitrogen were required to get the highest sugar yield. Wheat is different, less nitrogen residues, and therefore we have a more pronounced response and I would say wheat needs about 75 kilograms to get the same kind of response as after pea and sugar yield. But what I would like to focus more on is that if the maize is a previous crop, because of the problems with root growth and root infestation, it has a much weaker response to nitrogen, and we get a yield increase even up to 75 kilograms of nitrogen. So this shows that the root growth of the sugar beet is very much affected and is not able to take the nutrients and nitrogen, proxy for that, very efficiently, and therefore it needs to be compensated by some additional nutrient application in order to get the best response in the field. These trials have done very much with maize as a crop in rotation, because that's I think an easier crop to manage. But we have to bear in mind that's not only maize, which is a host plant, it's also soybean. And I think in Astata and also in other companies in Ukraine, you have also soybean in a rotation. I think what has been said about maize applies to maybe the same or to a similar degree also to soybean. So a rotation where you have both soybean and maize might respond to, in terms of disease severity, similar to what I have presented for a maize crop rotation. So the more host plants you have in a rotation, the higher is the risk of infestation and spread of disease, root rot disease. So crop rotation and crops in rotation is one very significant measure to control somehow Rizoctonia solani. But there are also other options to combat root rot. And in the next couple of minutes, I will talk about the effect of an intercrop and also about the effect of fungicides on root rot. And finally, of course, the choice of the variety. So intercrop, there is the opportunity to grow an intercrop in your rotation. Because I learned that you have a rotation where the sugar beet follows very much the winter wheat, and the harvest of winter wheat would allow a good growth of an intercrop like oil radish, Raphanus sativus, brown mustard, Brassica junctea, a rape type of crop, white mustard, and Phacelia. Those were the intercrops tested, and their effect on the disease severity of sugar beet grown after this intercrop, and the control has been a fallow crop, so which is normally the case in your system, where you have a bare soil after wheat has been harvested and the soil has been plowed until sugar beet are planted. So this trial has been done on two sites. One was more or less free of Rizoctonia solani, and on both sides we have a treatment which has been oculated and a non-oculated one. On the other side, we had a site which was already naturally infested. And then we had this the effect of the different intercrops. If we had a Rizoctonia solani free site, the difference between the different intercrops and the fallow was not significant. And also the response of white sugar yield, there was no response to the presence of an intercrop before sugar beet has been grown. The opposite applies to a site where we had a almost high pressure of Rizoctonia. So when we had a high pressure, either in the inoculated or non-inoculated treatment, we had an increase of the disease severity if the soil has left a fallow, bare soil from over winter, early spring, and small reduction in disease severity with these different intercrops. And in the inoculated, on the inoculated sites, so meaning with a high pressure of Rizoctonia root rot, we had a kind of significant response to the presence of different intercrops compared to the fallow treatment. So this white sugar yield was down with this high pressure to just 3 tons. And then with having an intercrop in place before sugar beet, the yield was in the order of magnitude of even 7 to 8 tons. So that seems to be a kind of significant effect of the presence of intercrop if the inoculation potential is very high on the site. Sometimes you find the inclusion of the intercrop termed as biofumigation, because by the intercrop you fumigate the soil and there are indications that there are direct effects on the Rizoctonia solani, but also some indirect effects can help to suppress Rizoctonia. So as I said, it works like you introduce an intercrop into the rotation, and it seems that like oil radish and brown mustards have a very good, amongst the intercrops, the best effects. So the objective is to produce a very substantial quantity of biomass in the intercropping period. And this biomass should be chopped and incorporated at start of flowering. So normally the intercrop shouldn't produce seeds. It should produce vegetative biomass. And from this vegetative biomass, the glucosinolates, a compound in these crops, are supposed to hydrolyze as isothiocyanates, a chemical compound which is supposed to be toxic to the pathogen. So this is the direct effect on suppressing Rizoctonia solani, and how this effect on yield can be explained. But there's also an indirect effect by promoting the antagonist in the soil of Rizoctonia solani, like Trichoderma and Pseudomonas, feeding on this organic matter. And of course, the way the soil structure and so on is also improved with organic matter, as long as it's evenly incorporated into the soil. Otherwise, of course, you can have the trade-off that even you increase the inoculation potential by the environment created by packed organic matter. Of course, as a trade-off, we have the cost for establishing and developing the intercrop, and we all know this crop has evapotranspiration. The transpiration to produce biomass is quite significant, and in areas where water is a shortage, maybe the trade-off of this kind of additional water consumption is too high to include it in the rotation for the sake of suppressing Rizoctonia solani. Another method to combat Rizoctonia root infestation is, of course, the use of fungicides. And to be honest, in a way, I don't know whether any fungicides for this purpose are registered in Ukraine. But I think it's anyway worth to share this information from research where they have tried the effect of two herbicides compared to a crop control. the herbicide has been atoxystrobin, Amistar extra. Probably that says something to you. At this rate, at 1 liter per hectare applied, and the combination of this one with the difenoconazole, again with this active ingredient, and 1 liter applied at the 6 leaf stage, and the canopy closure, 2 applications. So we have 2 sites again, the one with natural infection, and for the benefit of time, we can just conclude that the disease severity of different varieties being resistant, intermediate, or susceptible were not very much pronounced. Of course, the susceptible showed a bit higher disease severity, but when we look at the white sugar yield, the application of the fungicide hasn't caused any kind of significant yield effect. Here it was even more or less the opposite, so the control was higher compared to the treated one. But under conditions where we have very high pressure, and this may are considered as inoculated conditions, we see that the effect on a susceptible variety is very pronounced, and the effect comes only from the atoxystrobin. So the difenoconazole mixture was not required. So atoxystrobin Amistar extra has decreased the disease severity quite significantly, almost in all varieties. And also the white sugar yield has responded to the application of the fungicide. The most, of course, in a susceptible variety, the least in a resistant variety, but even under very high root rod pressure, we see an effect of the fungicide treatment. So in Europe, so far, there's no fungicide registered for control of root rot. I mean, in EU, I don't know really about Ukraine, to be honest. You probably know much better. But in US, this application is common practice with proven effectiveness at the cost of about 30 to 40 US dollar per hectare. So results from the US show this kind of picture as an effect of the Amistar extra treatment on the white sugar yield per hectare. They have tried different modes of application. It looks like a very early in furrow application, and application at the 6 to 8 leaf stage, even on a low rate, is the most effective application of this fungicide. And compared to a control, untreated control, the yield response to this fungicide treatments seems to be in the order of magnitude of 1 to 2 tons of white sugar yield, which is probably quite significant when you look at the economy of the cost for application, but the additional return you can get out of this fungicide treatment. It's just a question whether this is registered to be used, and of course the pressure of the disease in general. Another method to combat, of course, is the choice of the variety. In our briefing, you told me that you use KWS varieties or Struve-Diekmann varieties. This information comes from their brochures. You see, in a way, the features of these different varieties in terms of tolerance against different pests and diseases. The one of interest, of course, is the tolerance towards Rhizoctonia. Today, there is a KWS variety called Breda, which is supposed to be Rhizoctonia tolerant, and also has Rhizomania tolerance. And also Struve-Diekmann has two different varieties, what I could read from their brochure. Timu and Premier both seems to be Rhizoctonia and Rhizomania resistant, and have also some other features. I'm convinced that in a way you have all this information when you decide about purchasing varieties. This is just to highlight that these varieties exist, and from these suppliers I found these varieties which have this feature and can help very much on controlling Rhizoctonia. But of course it have some certain disadvantages. They are only tolerant to Rhizoctonia, not resistant. So yields are not guaranteed under extreme infection. So of course, in a way, when you have to deal with Rhizoctonia solani, you need to think about all kinds of measures to control it, and the variety is just one factor to deal with. So also tolerant varieties remain susceptible to the fungi and contribute to the building up of the inoculum, and they don't protect against this damping off early disease, which comes from a different Rhizoctonia strain. Tolerant varieties carry a yield penalty in healthy soil compared with standard Rhizomania material. I don't know whether this applies still for these varieties, that there is a yield penalty for the Rhizoctonia tolerance, but of course, it's always a kind of balance between what would the effect on controlling the Rhizoctonia, or in a way what kind of yield penalty we need to accept. Let me conclude my talk with some key messages we need to take on board to control or to reduce Rhizoctonia root rot. Crop rotation is important. A 3-year break from cultivation can help to reduce the inoculum and the disease severity. So no host plants should be grown in this break. Host plants such as maize, soybeans, ryegrass. Also, weeds should be consistently combated during the time in order to reduce Rhizoctonia solani pressure. Tolerant varieties should be considered and should be chosen on sites where we have known infestation and high infestation of root rot. Unfortunately, yield levels of these varieties can be lower when the soil is not, let's say, sufficiently infested than with standard varieties. All measures that promote the rapid growth of sugar beets can reduce the incidence. Good soil structure, evenly incorporated harvest residues, prevent soil compaction and waterlogging. Appropriate nutrient and lime supply strengthens the crop and contributes to the resistance of the crop. Also, we need to take care about that soil doesn't spill on the beet heads, because from the beet heads, during cultivation, also the disease can be spread. Intercrops like oil-radish, yellow or brown mustards and others can promote antagonism and have maybe also a direct effect, and also improve the soil structure, which gives a better kind of soil moisture pattern, which can suppress root rot to a certain extent also. With this, I'd like to thank you for your attention, and thank you for sharing your precious time with me on this topic. And I hope in a way you get some ideas what might apply in Ukraine, and maybe some ideas what you may can change under your condition.